Automated fluorescence sequencing. Since the completion of the Human Genome Project, technological improvements and automation have increased speed and lowered cost to the point where individual genes can be sequenced routinely and can sequence well over 100,000 billion bases per year, or an entire genome can be sequenced. Since the completion of the Human Genome Project, technological improvements and automation have increased speed and lowered cost to the point where individual genes can be sequenced routinely. One new sequencing technology involves watching DNA polymerase molecules as they copy DNA, the same molecules that make new copies of DNA in our cells, with a very fast movie camera and microscope, and incorporating different colors of bright dyes, one each for the letters A, T, C and G. This method provides different and very valuable information than what's provided by the instrument systems that are in most common use. Another new technology entails the nanopore based DNA sequencing. Involves threading single DNA strands through extremely tiny pores in a membrane. DNA bases are read one at a time as they squeeze through the nanopore, and the bases are identified by measuring differences in their effect on ions and electrical current flowing through the pore. The DNA to be sequenced is prepared as a single strand or template. DNA is supplied with a mixture of all 4 normal deoxynucleotides in ample quantities. They are DATP, DGTP, DCTP, DTTP. A mixture of all 4 dideoxynucleotides, each labeled with a tag that fluoresces a different color, each present in limiting quantities: DDATP, DDGTP, DDCTP, DDTTP and DNA polymerase 1. Because all 4 normal nucleotides are present, chain elongation proceeds normally until by chance DNA polymerase inserts a dideoxynucleotide shown as colored letters instead of the normal deoxynucleotide shown as vertical lines. If the ratio of normal nucleotide to the dideoxy versions is high enough, some DNA strands will succeed in adding several hundred nucleotides before insertion of the dideoxy version halts the process. At the end of the incubation period, the fragments are separated by length from longest to shortest. The resolution is so good that a difference of 1 nucleotide is enough to separate that strand from next shorter and next longer strand. Each of the 4 dideoxynucleotides fluoresces a different color when illuminated by a laser beam, and an automatic scanner provides a printout of the sequence.